Hello everyone, and a warm welcome to this webinar on the topic Vaccine Manufacturing Facility Qualification. This webinar is a part of series of planned webinars with the goal of providing information on available career options in the field of vaccinology. My name is Clive Nyaribo, Assistant Research Officer at KEMRI-Wellcome Trust Research Program, providing support to the Vaccine Workforce Development Program. Together with my colleagues, Professor George Warime, Marianne Munena, and Ravenda Navura, we welcome you to this session. Before we get started, a few housekeeping matters. Kindly take note that this webinar is being recorded. Kindly feel free to use the chat option to share your questions during the presentation, and we'll address them during the Q&A session. We'll have a few open slots for people to directly ask questions to the presenters. If your internet is stable, you can raise your hand using the available icon. As Arya mentioned, these are series of planned webinars with the goal of providing information on available career options in the field of vaccinology. This being run under the KEMRI-Wellcome Trust Research Program, which is a world-renowned health research unit of excellence based in Kilifi and Nairobi in Kenya, as well as Mbare in Uganda. Over the years, we have excelled in the use of novel ideas and collaborations to achieve better health for Africa, while also providing African scientific leaders. Our vision of the program is to take cutting-edge and novel research relevant to national, regional, and global needs. It's under this program that we have the Vaccine Workforce Development Program that aims to expand the technical know-how and capability for sustainable end-to-end vaccine research and development in Africa. If you can be able to hear me well, kindly. Okay, great. It's under this program that we have the Vaccine Workforce Development Program that aims to expand the technical know-how and capability for sustainable end-to-end vaccine research and development in Africa through training and knowledge exchange activities in the vaccine ecosystem. This involves areas such as target product profiling, research and discovery, preclinical and clinical studies, biomanufacturing, regulatory affairs, and deployment. The workforce development through 4 channels: internship at vaccine manufacturing institutions that aim at establishing a mentorship scheme that links interns with senior vaccinologists and healthy stock market systems, and engagement with academics globally to share and strengthen experience in vaccine research and development and manufacture and enhance career development. 2, a full funded master's level course in vaccinology where applicants are selected based on their direct involvement in vaccine research and their professional excellence and academic achievement. Webinars like what we have today to area career scientists, graduate and undergraduate university students in Africa to provide information on possible career options for university students for informed career decision making. 3, 4, Vaccinology School Engagement Program where students from local secondary schools are invited for lab tours and career talks to spark their interest in science, especially in the vaccinology field. This program is in alignment with the Africa Union's platform for harmonized African healthy products manufacturing, ambition to increase local vaccine production to meet 60% of Africa's routine immunization needs by 2040, and strengthen the capability for vaccine research, discovery, and manufacture in Africa. And this webinar series is part of these engagements. Today, we are glad to have John Wassily, who will be discussing the topic vaccine manufacturing facility qualification. John Serim Wassily is the head of Qualification and Validation Department at BioGeneric Pharma, Cairo, Egypt. John holds a Bachelor of Science degree in Pharmaceutical Sciences from Faculty of Pharmacy, Cairo University. With over a decade of hands-on experience, John is a recognized expert in establishing and governing validation systems for complex, sterile manufacturing environments, particularly within greenfield and brownfield projects. John excels at developing robust organization strategies covering the full validation qualification lifecycle, from concept to commercial operation. John's key contributions include strategic system establishment. He is proficient in establishing facility-wide quality frameworks, including the validation master plan, calibration master plan, and comprehensive environmental monitoring. Risk assessment for clean rooms. John led the AU GMP Annex 1 gap analysis project at the sterile facilities, securing AU market export licensing. He has successfully prepared sites for and passed multiple stringent international audits, including the EU Initial Licensing Audit 2019 and the WHO MPP Assessment 2022. New product introduction. John actively supports new product introduction teams by issuing project timelines, allocating resources, and setting smart action plans to efficiently drive validation and qualification activities from inception through to product launch. John has hands-on experience or expertise in validation and qualification areas, including sterile process validation, developing and executing strategies for aseptic process simulation, including process validation and clean validation. Facility and utility qualification, leading qualification activities for HVAC systems, water systems, and thermal mapping for equipment and storage areas. QMS and compliance, authoring essential general SOPs directing quality risk management projects and conducting gap analysis against global regulatory standards, and also technical documentation. Over to you, John.
Hi, Clive. Thanks, Clive, so much. And hello, everybody. Do you hear me well?
Yes, we can, sure.
Okay, and you can see my screen?
Yes, we can, sure.
Okay, very good, very good. I'm so glad to be here with you today and presenting this topic, and hope you get the most benefit of it. Let us start immediately. Our topic today will be covering the vaccine manufacturing facility qualification. And this will be a quick guide and an introductory session about the qualification for the vaccine manufacturing facility. Okay. First of all, let me introduce you some terminology that I want you to get familiar with. Sorry. Okay. Do you see my screen now, right? Okay. Very good. Okay, so we can proceed. First of all, I want you to get familiar with some basic terminology like the validation and qualification. The validation and qualification are usually interchangeable terms, and they are used in order to make sure that equipment, utility, system, and processes actually do and perform what they are supposed to do and perform in a consistent manner. By this, for example, if I'm talking about a sterilizing autoclave, so its qualification will be about to make sure that this sterilizing autoclave is actually performing and doing what it's supposed to do. It's actually giving us a product that is sterile and free from any microbiology or bacterial traces. So by qualification and validation, I mean that to make sure that the equipment, utility, system, or process is actually doing and performing as per the estimated and expected performance in a consistent manner. So every time that I will be operating my sterilizing autoclave, by doing some testing on it, I will be sure that finally it will give me a product that is sterile and free from any microorganism. Okay. Also, I want you to get familiar with the term of calibration, and you might encounter the term metrology. It's also known as metrology. This is to make sure that the measurement, either the measured and displayed values by any system, equipment, or instrument, is accurate and precise. So, for example, if we will apply this on the scope of a sterilizing autoclave, calibrating its temperature sensors is to make sure that the temperature sensor is actually displaying and measuring accurate and precise measurements. This is done normally by comparing the measurement device readings to a known reference standard and detecting any drifts and adjusting the accuracy and precision of measuring instruments. Okay. Here I want you to differentiate very well between the validation and qualification terms. Yes, they are interchangeable terms, but finally, they share the main concept but they differ in the application. For example, validation is establishing documented evidence that provides a high degree of assurance that the process, method, or system will consistently produce a product meeting its predetermined specs and quality attributes. The qualification term is sharing the main concept, but here it applies on utilities, equipment, and facilities. So we can say finally that validation term applies on the manufacturing process, cleaning process, sterilization process, aseptic process, gowning process, visual inspection process, analytical method, and systems like the computerized systems. So any process, in order to make sure that it's performing as expected, we will refer to this by saying validation. And on the other hand, qualification is about making sure that the equipment, devices, instruments, premises, and facilities, also utilities, is acting and performing as it's expected to perform, okay? But on the level of equipment, instruments, facilities, and premises. So both are sharing the main concept and the same concept, which is about to ensure that whatever process or whatever piece of equipment will actually perform what it's expected to perform. So, for example, by saying cleaning process verification or cleaning process validation here, I mean that to make sure that the cleaning process is actually giving us an equipment or a facility that is really clean. And by saying, for example, sterilizer autoclave qualification, as mentioned, is to make sure that the autoclave will give me a product sterile and free from any microorganism. Applying this on any other equipment, like the filling machine equipment that will be used in filling the vaccine products, will be about to make sure that the filling machine will finally give me the nominal volume that is expected to have in my vaccine product in a consistent manner, okay? Also, I want you to be very acquainted with the relationship between calibration, qualification, and validation. Finally, calibration is part of the qualification and qualification is part of validation. This is because I cannot rely on, for example, a sterilizer autoclave before making sure that its critical sensors, like the temperature sensors, are calibrated. And then I can go for its qualification in order to make sure that it will consistently produce a product that is sterile and free from any microorganism. And finally, I can use this qualified sterilizer autoclave in the process, the manufacturing process. And by performing a process validation, I can have finally a validated process that has been using a qualified equipment, and the qualified equipment was calibrated for its critical sensors. So this is mainly how the interaction and relationship between the different terms are occurring. Let us go forward towards the roadmap towards a qualified and operational vaccine manufacturing facility. By saying a manufacturing facility is qualified, it's not about a single activity of qualifying the facility only. It's about going through a roadmap constituting of various qualification and validation activities. It's about qualifying and validating all the supportive systems and equipment, starting from the QC labs with their instruments, to the production area with its equipment, also for the personnel that will be working in this manufacturing facility, and also qualifying the critical utilities that will be used in this manufacturing facility. mainly the qualification for the critical equipment goes for the direct contact and direct impact systems like the HVAC system, the water system, and the gas system. So in order finally to have a vaccine manufacturing facility that is qualified, it's about going a very huge roadmap, passing through different qualification activities of various systems and equipment. So putting all those systems and equipment like a pieces of puzzle together, we will have finally a qualified manufacturing facility that could be used for operational purpose and for vaccine manufacturing in a confident way. Let us see together how this roadmap can start. First of all, we can start this by listing all the different areas that will be under the qualification in our facility. So for example, listing that our vaccine manufacturing facility is consisting of 3 production lines, a QC lab, and utilities building. By listing all those areas, we will get acquainted and we will be aware that all those areas shall pass through different qualification activities, and covering and fulfilling all those qualification activities for all these areas will enable us to finally have what we call a qualified vaccine manufacturing facility. So for each of the listed areas here, we shall proceed in premises qualification for each area, for each individual area, utilities qualification, and equipment qualification. Let us start by the premises qualification. It's also generally known as facility qualification. By premises qualification, we mean, for example, if we will apply this term on the production lines, so we will list all the specifications that are listed in our room data sheet, okay? And we will start to do like a checklist for each room that is present inside this production area. So for example, for each room, we will check the room dimensions, we will check the HVAC related items, the number and type of supply and return air grills, the HEPA filters and their grades. We will check the construction and finishes. All those are specifications for the rooms, for the areas that should be well listed in a room data sheet. And our role here in the facility qualification or premises qualification for each area is to make sure that all the specs listed in the room data sheet are well checked and well fulfilled. Passing through the floors, the walls and ceiling finishing materials, the drainage system, if any interlocking system has been requested to be present inside the production area. Also checking the illumination level and allowed noise level for each room. Checking the room utilities that were requested in the room data sheet, either purified water, water for injection, pure steam, chilled water. Also checking any safety features like fire detectors, fire sprinklers, emergency exits, and so on. Checking also the electrical requirements in terms of UPS sockets, if required, and voltage supply. And finally, checking the temperature, relative humidity, and pressure monitors if they were requested in the room data sheet. So what will be requested in the room data sheet, the premises qualification will be about to make sure that all those requirements are well fulfilled for each room. This is just a very simple representation of how a premises qualification and facility qualification goes. Then proceeding to the equipment qualification. Let us imagine that we are currently talking about the production area. So we've covered its premises qualification, and now we will cover its equipment qualification. And that's what we've mentioned in the previous slide, that we will list all the areas that will be in the scope of our facility, and we will pass through their facility qualifications, their equipment qualifications, and their utility qualifications. So here we are addressing the equipment qualification, for example, of the production area. The same will apply for all the areas that will be listed. For the equipment qualification, it's not a single or simple activity, it's actually a life cycle. And here I'm presenting to you a process map for this life cycle. It starts by performing a system risk assessment, performing a critical attributes and critical aspects risk assessment, listing all the aspects that are critical to the end product quality and patient safety. So by listing all those aspects, we'll be able to start drafting our user requirement specification for that piece of equipment. The user requirement specification is a piece of document in which we will list all our requirements as a user, okay? All the specifications that we as users need to be fulfilled in our equipment that we are going to purchase, okay? And then we'll perform system classification, and we will see together through the next slides how to perform the system qualification and how we can make use of this classification, okay? And finally, here in this stage, which is the defining of requirements stage, we will perform the design specification. The design specification is actually that the supplier will draft and will issue a document to say that here are all your requirements as users. Here are all your requirements. They were fulfilled if we are talking about functional requirements, hardware requirements, or software requirements. So the supplier issue a document from his side and say that all your requirements that were listed in your URS have been fulfilled. And then going to the next stage, which is the design stage, we as end users will start doing the design qualification for the piece of equipment that we've purchased. The design qualification will be about comparing the specifications that we've requested in the URS document against the design specifications that were issued from the supplier side. And doing this comparison, we will make sure that all the requirements that we've requested initially were actually fulfilled. And then we will go to the validation planning. The validation planning will rely on the system qualification that we've performed in the previous stage, in the previous step. Based on the system classification, we will identify the qualification phases and the qualification testing to be performed. And we will see this together through the next slide. And then we will go for the qualification testing. The qualification testing is about going through different qualification phases: installation qualification, calibration, and operational qualification. It's about performing tests on the piece of equipment that we've received in order to make sure that it's well installed and it's well operating as per the initial specs that we've requested. Also, we shall address a document called the Requirement Traceability Matrix. The Requirement Traceability Matrix is about comparing the user requirement specifications to the design specification document and make sure that our requirements are not only well considered by the supplier in the design specification document, but also that they are tested in any of the qualification testing phases, either in the IQ, OQ, or PQ. And finally, we go for the acceptance and release for the piece of equipment. This will be by performing PQ testing, performance qualification testing, and I will take you through how this testing will be performed. This is the final stage, which is the release stage. By reaching this stage, here I'm releasing the piece of equipment for routine operation and for routine use in the vaccine manufacturing. And then periodically, I shall pass through this piece of equipment, making sure it's maintaining its initially qualified state by performing periodic review and monitoring. This is a quick and brief overview for the equipment qualification life cycle. I know it's a bit dense, but this is how the equipment qualification should go. Here I'm presenting to you the same stages that we've presented through the previous slide. First of all, the validation master plan here is the overall organizational approach to system validation, including the deliverables, personnel, testing strategy, and general acceptance criteria. This is the main umbrella document under which I will be stating all the qualification activities that should be done for each piece of equipment or any system. Okay? And then going through the URS, the user requirement specifications that we've presented through the previous slide. In this, we will define all the requirements that we need as users to be fulfilled through the piece of equipment that we are going to purchase. The requirement traceability matrix is a document in which we will make sure that our requirements that were listed in the URS document are well considered in the design specification document that was issued from the supplier and are well tested as well in any of the upcoming qualification testing phases, either DQ, IQ, OQ, or PQ. For the Q, as mentioned before, it's about performing a checklist in order to check that all the specs that were listed in the URS were designed and well considered by the supplier. So this is the design qualification to make sure that the design that has been issued by the supplier is well considering all the requirements that we've listed in our URS document. For the installation qualification testing, it's about that I've received the piece of equipment finally on site in the vaccine manufacturing facility. So I will make some testing to make sure that it's well installed, that all the components are there in their places, that all the utilities are connected to this piece of equipment, that also I've received all the related documentation and manuals. And finally, the last stage of the IQ testing will be the calibration, which is about to calibrate the critical sensors and critical control items before performing the operational qualification testing. Then going through the operational qualification, I will start doing some testing in order to make sure that the piece of equipment is well functioning. So if here, for example, I'm talking about an autoclave sterilizer, so I will make sure that all the sterilization recipes and programs are well operating and all the sterilization phases are progressing smoothly without any abnormalities or any interruption in the sterilization phases. Also, part of the operational qualification is to make sure that any alarms are triggered as per the user requirement specification. For example, I may have asked the supplier to trigger alarms upon reaching certain set points or upon exceeding certain limits. So in this phase, it's my responsibility to test that the alarms are well triggered. Finally, the performance qualification is about making sure that the piece of equipment is actually performing consistently on the real-time and live product. So for example, if I'm talking about an autoclave sterilizer as an example, the PQ here will be to put an actual load to be sterilized and see how the autoclave will be performing. If the autoclave will finally yield a product and a load sterile and free from microorganisms or not. Also, part of the PQ is to challenge the extreme and the worst case conditions. So for example, if I'm talking about a PQ for a vaccine filling machine, so here I shall be challenging and I shall be testing the extreme filling speeds of this machine in order to make sure that it's well performing on the extreme conditions. And finally, I will issue what is known by the validation summary report. It's a report concluding all the qualification testings that have been done on my piece of equipment and putting a conclusion that now the equipment is released for operational use and can be used normally for vaccine manufacturing. Also, I want you to get familiar with another representation for the equipment qualification life cycle. It's usually presented in the regulatory guidelines by the V model. The V model here is presenting the same qualification phases and the same qualification testings that we've explored together in terms of user requirement specification, design specification, design qualification, installation qualification, operational qualification, and performance qualification. But here it's presented in a V model shape in order to build a correlation between each phase and how it's correlated to the previous phases. This is just for you to be familiar with how the equipment qualification life cycle could be presented in the regulatory guidelines. Also here, I want you to be aware with the qualification and validation documentation. I've mentioned for you the validation master plan, and I've mentioned that this is the high-level document, the main umbrella under which I will be putting the plan and the main aim for the validation phases and for the validation project, addressing all the validation and qualification activities that might take place in my vaccine manufacturing facility in order to have finally a qualified facility for vaccine manufacturing. So here I'm showing to you all the documents that should be issued. It's not only about that you will perform qualification testing, it's also about that you will issue a document to act as a documented evidence and proof that the qualification testing has been performed. So first, you will issue the validation master plan, and then you will have to issue a general SOP for each qualification or validation program. And then you are assumed to issue a very specific qualification or validation protocol before the execution of that protocol. The protocol here will be directing you to pass through which tests to be performed specifically on this piece of equipment or this process, and then you will be able to proceed and execute the validation or qualification activity. And finally, you will be requested to issue a final report in order to conclude all the qualification testing results and to have a final conclusion saying that the process or equipment is now valid for operational use. After this, if any changes have happened on your already qualified or validated system, equipment, facility, or utility, you will be assumed to issue what is called a change control in order to list all the changes that might have happened on your equipment or systems. For example, introducing a new critical sensor on your piece of equipment. So this is a change that will necessitate a change control in order to assess the impact of such an introduction on your already qualified piece of equipment and to decide whether this change will necessitate you to requalify or to revalidate the already qualified or validated system or not. So this is for you to figure out how the qualification or validation documentation should be proceeding. Going through the system classification or categorization, if you can remember, I've listed and mentioned to you that after issuing your user requirement specification, you shall be categorizing or classifying your system or your equipment. This classification or categorization will help you to identify the extent of qualification. So I've mentioned to you many phases and many testing that are assumed to be performed as part of the qualification lifecycle. So am I assumed to cover all those testing phases and all those qualification testings? No, you will not be asked to do and to cover all those testings. You will have to categorize, and based on this categorization or classification of your equipment, we will identify and determine the extent of qualification testings needed. So for example, taking an example of the QC lab instruments and how the regulatory guidelines like the United States Pharmacopoeia is categorizing and classifying the QC lab instruments. In the USP, it's categorized into 3 groups, Group A, B, and C. And as you can see here, every group will mandate a certain level of qualification to be performed. So for Group A, for example, it's the simple and least complex instruments. And based on this, no further qualification will be needed. It's only about doing simple observation. For Group B, it's a more complex instruments and equipment like the pH meter and the oven that we'll be using in the QC lab. And based on this, it's required more sophistication of your qualification testing, like, for example, requiring only routine calibration, maintenance, or performance check. And reaching the highly sophisticated and complex QC lab instruments like the HPLC and the spectrophotometers, here are significant degree of computerization and complexity. So we shall undergo full qualification and software validation, passing through all the qualification testing phases, starting from the URS, IQ, OQ, PQ, and all the phases that were mentioned on the previous slides. Here I'm presenting to you other example for the classification and categorization of other equipment. Here will be the equipment related to the vaccine production area and utilities equipment. It will be based on 2 categorizations, and the multiplication of those 2 categorizations will yield a specific color, and based on this color We will identify the extent of testings that shall be performed on each piece of equipment before being released for operational use. So for example, based on the GMP impacts here, we will identify that we will have either a class 1 equipment, class 2 equipment, or class 3 equipment based on the impact of those equipment on the product quality and patient safety. So for example, for class 1, those are equipment that doesn't have any impact on product quality or patient safety. For class 2, those are equipment that have an indirect impact or that are supporting the direct impact systems, like the chiller systems, for example. And for class 3, those are the most complex as they are having a direct impact on the product quality and patient safety, like the filling lines, the isolators used in vaccine manufacturing and filling, and the sterilizer autoclaves. Okay? Multiplying this by the complexity of the software used on those equipment, as per the ISPE guideline, it's categorized into 4 categories, category 1, 3, 4, and 5. Based on the software complexity, we will have a color-coded result. Based on this color code, as you see here on this slide, we will identify the level of sophistication and of qualification needed for each piece of equipment. Okay. So now that we've finalized the part of facility or premises qualification and then equipment qualification, now we will be exploring the part of critical utilities qualification. Those are the qualifications related to only, for example, one area of the listed areas in the very initial listing. If you can remember, we've listed that we are having 3 production areas, 1 QC lab area, and 1 utilities building area. So we took an example, for example, of the production. And for this production area, we've seen how to perform the premises qualification, how to perform the equipment qualification, and we've seen that the equipment qualification is not a simple activity, it's actually a life cycle. And now we will be exploring together the utilities qualification for that production area, for example. Here I will be showing to you the HVAC system qualification for a production area. By saying HVAC system qualification for a production area, the aim of the HVAC system qualification will be to make sure that the clean room in which I will be preparing, formulating, manufacturing, filtering, or filling the vaccine is actually meeting the predetermined and designated specs for the cleanliness level. So, for example, for a preparation room, for a formulation room, for a vaccine, the conventional used cleanliness grade is grade C. So, by qualifying the HVAC, I will make sure that the HVAC system can meet the cleanliness grade of grade C. This is by using particle counters and measuring the concentration of particle inside the clean room. So for example, I will measure the concentration of particles who are corresponding to size 0.5 micron and 5 micron. I will measure them at rest state, and they shall be, for example, meeting this limit. If they are exceeding this limit, this will mean that the clean room is not meeting the cleanliness grade of C, and it's not allowed to be used for manufacturing or formulating the vaccine that I'm talking about. The same apply for all the grades, but here I want to drag your attention to a very important point that upon increasing the cleanliness grade from C to B to A, going to the A, this is the highest level of cleanliness grade, and this is actually where the vaccine filling takes place. And you can see here that under grade A, we are having the least number of allowed particle concentration for the size of 0.5 and 5 micron. Okay, so by qualifying the HVAC system, I will be releasing the HVAC system for routine use and routine operation with a high degree and level of assurance that it will enable me to have a clean room with the needed cleanliness grade. Here I'm presenting to you a summary for the HVAC qualification or clean room validation testings that are to be performed in order to say that my HVAC system is fully qualified and is released for operational use. It's not only about performing the particle measurement for the sizes of 0.5 and 5 microns as mentioned on the previous slide. It's also about performing filter integrity testing for the filters that are installed in the clean room itself to make sure that the air is filtered and meeting the cleanliness grades that we are talking about. And here we are listing all the acceptance criteria that we shall meet for every test. By, for example, mentioning the containment leakage test, this is a very important test in order to prevent any cross-contamination, especially if we are talking about a vaccine manufacturing facility, because the vaccine is usually a hazardous product, and I don't want any escape from the less pressurized room or the hazardous room to the surrounding or adjacent rooms. So by this test, I make sure that the clean room in which I'm manufacturing my hazardous vaccine product is tightly sealed with no leakage, and no leakage is present from the negatively pressurized rooms onto the positively pressurized rooms. Going through the other tests, either recovery rate test, this is to make sure that in case of any unintended dirt that has been introduced onto the clean room, my HVAC system will be able to recover the initial cleanliness grade within a specific time frame, which is usually not exceeding 20 minutes. So this test is performed by intentionally generating particles inside the clean room and see, by observing the particle count, how the HVAC system will recuperate and will recover the initial cleanliness level. Also important is to perform an airflow visualization or smoke test. This is to visualize the airflow and to make sure that the air is flowing from the highly pressurized rooms, which are usually the rooms having the highest cleanliness grade, like the grade B, for example, to the lower pressurized rooms, which are rooms of less cleanliness grade, like grade C. So it's important for you to know that by having a higher cleanliness grade, also I will be having a high pressure inside this room and this grade. And the air is expected to be flowing from the highest pressure to the lowest pressure, or from the highest cleanliness grade to the lowest cleanliness grade. Also, this test should be performed at rest and in operation in order to make sure what is the impact of any movements or any operating personnel inside the room, how they might impact the airflow inside this room. Also going through the non-viable particle count, this is the test that I've been talking about on the previous slide, which is about to measure the concentration of particles that are corresponding to the size of 0.5 micron and 5 micron, because by measuring those particles, you will be sure that you are meeting the needed and the required cleanliness grade. Also, it's important to measure the viable particle count, not only the non-viable, the viable in terms of microorganisms, in order to quantify the viable microorganisms. And here we are having also certain limits that we shall meet for every grade using different sampling methods. Also, part of the HVAC qualification testing is to measure the differential pressure, to measure the airflow volume, to measure the temperature and relative humidity. And as you can see, for each of those tests, we have acceptance criteria that we shall meet in order to say that, yes, my HVAC system is now fully qualified and could be released for operational use. Here, I'm displaying to you the frequency by which the HVAC qualification testing and the clean room validation testing should be performed. And because this is a critical impact system, this is a critical utility, it should be performed by a certain frequency. As you can see here, for each class, we are listing all the testing, the qualification testings that should be performed and with which frequency they are expected to be performed. Because it's not only that we will perform the qualification initially. If you can remember the diagram of the life cycle of the qualification, it's also repeated periodically in order to build confidence that the initially qualified state for the utility, for the equipment, and for the systems in general are always maintained throughout the entire life cycle of the system. So periodically, they shall be tested either here as per the ISO every 6 months or every 12 months. Okay, going through the other critical utility qualification, which is the water system, an integral part of the vaccine manufacturing qualification is to qualify the water system that is being used in the formulation of the vaccine. And here we are having the water for injection used as a primary raw material in vaccine manufacturing. And you shall be very well aware that the qualification here is a life cycle and it's a time consuming. It's consisting of 3 phases. Phase 1 is about 2 to 4 weeks. And in this phase, extensive daily sampling for all the water for injection use points take place. And all the samples are tested for the below tabulated tests. And each test shall meet the tabulated acceptance criteria. The same will take place on phase 2, which is 2 to 4 weeks as well. Here, the sampling frequency could be with a reduced frequency. And then phase 3 is about sampling your water for injection system for a full 1 year. And this is to capture any seasonal variation that might occur due to the winter and summer variation, because the winter and summer variation could impact the microbial load. And it's very important to cover this through your water system qualification. Okay, so now we finalized the first part of the roadmap towards a qualified and operational vaccine manufacturing facility, which is listing all the different areas under qualification. And for each area, we will cover the equipment qualification, the facility or the premises qualification, and the utilities, the critical utilities qualification. Going through the next point, which is preparing a validation matrix for each area. Validation matrix comprising the premises, utilities, and equipment. The validation matrix here will aim for you to be following up your activities in order for you not to drop any single activity and to make sure that all the qualification activities have been fulfilled and have been met. So for example, this could be a general example for a validation or qualification matrix for the premises. You will just simply list all the rooms that are present inside a given production line, and using a color coding, you will assign the premises qualification status. This is for you and for all the stakeholders inside the manufacturing facility to visualize the qualification status for the premises, for example. The same, you can do it and perform it for the utilities. For example, if we are talking about the HVAC system, so we are going to list all the air handling units that we are having supporting a given production area. And we are going to use the color coding in order to assign the IQ, the calibration, the OQ and PQ for each of those air handling units. Also, you may use this general example of validation and qualification matrix for the equipment. So for example, covering the equipment of the production area, you can list all the equipment that you are having on your production line, their codes, their locations, and using a color code, you can assign the status of your equipment qualification for all the qualification testing phases and life cycle. Okay. This was the first pillar for having a qualified vaccine manufacturing facility. The second pillar is about having well-trained and qualified personnel. And here, if we are going to explore the guideline of PDA Technical Report 22 that has been recently published, it's well mentioning that your personnel that will be working in your vaccine manufacturing facility shall pass through a very extensive training and qualification program. Sequentially, level 1 qualification will be in order to allow them to enter into the clean room. So they shall be trained firstly on the general SOPs, reading the general instructions. They shall attend training sessions regarding the basic hygiene, basic microbiology, sterility assurance, the gowning procedures, the contamination control. And finally, in order for you to assess if they were qualified and passed successfully the level 1 qualification, you can do a sort of assessment, theoretical, or by observing their gowning qualification and clean room behavior. Once they pass the level 1 qualification, we can proceed to the level 2 qualification, which is a training on job, and it will be a role specific. Okay. And then passing the level 2, you can proceed into the level 3, and it will be for you to observe them in the aseptic course in the sterile clean rooms and observing their aseptic behaviors, okay, and how they perform inside a clean room. And the final level of qualification or training for your personnel will be to periodically and on an ongoing basis perform a training for them as a sort of refresher training and assess this by observing their participation in different activities like the MediaFel activities that we will see together eventually through the next slides. So here I'm aiming to inform you that an integral part of your vaccine manufacturing facility qualification is to have a well-trained personnel and qualified personnel. Also, it's important to cover the process validation aspects. If we are talking here about the gowning process, it's important to cover the personnel gowning process and to make sure that the personnel have been qualified on how to perform the gowning procedure before being allowed to enter the clean rooms and the sterile cores, and most importantly, the Class B filling rooms, which are the highest grade cleanliness rooms in which usually the filling of vaccines takes place. So part of the personnel training and qualification is to qualify their gowning procedure and to what extent they are competent in performing the gowning process. So for example, here I'm going to present to you how for a newcomer the initial gowning qualification should take place. First, the personnel shall attend all the assigned training sessions. Once attending them, he will be asked to perform 3 gowning qualification runs. And by gowning qualification runs, I mean that the personnel will be asked to perform the routine gowning process, and microbiologically, some samples will be taken from the gown that he has put in order to make sure that he is competent enough in putting on the sterile gown without compromising its sterility. Because with this sterile gown, he's going to access the sterile cores and he's going to deal with the vaccine filling in grade B environments, okay? And then after performing the gowning qualification and after proving his competency and after passing successfully the microbiological sample for the gown, he can attend a MediaFel simulation. And the MediaFel simulation, we will see together what it's about through the next slides. Passing through all those steps, he can be assigned a qualified status and he can access the sterile cores and could be allowed to enter the aseptic Class B cores and to deal with the grade A environments usually taking place under the laminar air flows, okay? The same takes place when we are talking about a requalification for a personnel, because it will not be only about an initial qualification for that personnel, but it will be about requalifying him periodically on an annual basis by repeating the qualification programs that we've mentioned, by repeating the training session, but by repeating the microbiological sample, by repeating his attendance to a MediaFel batch, and so on. And here, to be more practical, this is how the gowning qualification takes place. If you can see here, this is the exact gowning for a Class B area for the sterile and aseptic cores. So the personnel shall wear and shall put on the sterile gowns, like in this picture, and microbiologically, we will take samples from the mentioned locations. Why those locations specifically? We are usually sampling the locations that are directly contacting or could be directly contacting the product or could be directly facing the working level. So the red ones are the most risky areas to be sampled, from the personnel gown. Those are the less critical, and the green ones are the least critical locations, but are also to be sampled, because by sampling these areas, we will see to how extent the personnel have been competent to put on the sterile gown without touching or compromising the sterility of the gown itself. Also, part of your preparation for the vaccine manufacturing facility qualification is to qualify the procedure and the process by which you render your clean room decontaminated, disinfected, and sanitized. This is usually done following a process called the dry fog. The dry fog is the usual followed process for cleaning and decontaminating the highly classified areas like grade B and A areas. And this is to make sure that we've reached the needed sterility assurance level. And we will be sure of that by taking environmental monitoring samples from the areas after being decontaminated. And also, we will make sure of that by placing a challenging biological indicators specifically designed for the assessment of the dry fog process. Okay, and the biological indicators here will be about some spores of Wassily styrothermophilus, which are the most resistant to the decontamination using the dry fog process or the hydrogen peroxide process. And we will make sure that the spores inoculated inside those biological indicators have shown not less than 4 log spore reduction. By achieving this, we will make sure that our dry fog process using the hydrogen peroxide is now able to be used for routine decontamination, sanitization of our high-grade rooms like grade A and B rooms. Here I'm presenting to you a quick overview about how the dry fog process takes place. It's about having a tank, a portable fogger device or a tank, and a compressed air source. And in this tank, we will put the sterilant solution, which is a mixture of the peracetic acid and hydrogen peroxide. This sterilant solution aims to decontaminate the grade B and A areas because it's effective against all forms of microorganisms, either bacteria, spores, and viruses. And then using the compressed air, we will spray and fog the sterilant solution that is present inside the tank through a head constituting of nozzles. Here, the nozzle number and direction is very important parameter because based on those nozzles, we will have a fogged solution of atomized droplets of the sterilant solution, the size of which will be approximately 7 microgram, 7 micrometer. And these droplets will act as a dry fog procedure, decontaminating even the hardest to reach areas within the clean rooms and the grade A and B areas. After the dry fog program ends, you can use the room normally with no additional need for surface cleaning because the mixture of peracetic acid and hydrogen peroxide is fully biodegradable and non-corrosive. It finally degrades into water, oxygen, and acetic acid. Okay, and after the end of this program, you can sample the room microbiologically to make sure that yes, the area has been meeting the requirements of grade A and B requirements. This is a representation for the actual dry fog process and how it actually takes place. Here, as you can see, the red and purple locations are the locations of the dry fogger device or the dry fogger tank in which we will be placing the sterilant solution. And the arrows are representing the nozzle number and directions that will be spraying the sterilant solution within the area under decontamination or sanitization. And the numbers here encircled are actually the locations of biological indicators that we are going to challenge the dry fog process with. So we are going to put biological indicators constituting spore-forming Geobacillus thermophilus microorganisms, and we will start the fogging process on them, and then we will collect by the end of the program those biological indicators in order to make sure that the process has been efficient in killing all the microorganisms and the challenging biological indicators. Usually, it's important for you to know that those locations have been selected based on a risk assessment. We are covering the worst case locations, as you can see here in the table beside. The final and the end part of our vaccine manufacturing facility qualification is to reach the aseptic process simulation stage. This is the end point of our facility qualification. Usually, you shall be aware that the vaccine is a protein-based product. So in order for us to produce a vaccine and to manufacture a vaccine that is rendered sterile and injectable, we are following usually two approaches. Either upon producing and filling this vaccine to terminally sterilize it in an autoclave, which is not applicable in the case of vaccine because the vaccine will normally denature and degrade inside the autoclave, or to follow the approach of aseptic processing, and this is the approach that we shall be following in case of vaccine manufacturing in order to produce a sterile and injectable vaccine product. So the aseptic processing is about having sterilized containers, sterilized, for example, vials or pre-filled syringe or whatsoever the container in which the vaccine will be filled, okay? And having sterile product, which is rendered sterilized by filtration, and then filling them all together under grade A aseptic filling conditions. And here, the aseptic filling conditions of grade A are rendered aseptic and sterile after fulfilling all the qualification testings that we've mentioned for the clean room and for the laminar airflows and for the dry fog process itself, which was used in order to decontaminate the clean room itself. And here we shall perform what is called an aseptic process simulation. The aseptic process simulation will be about simulating the exact manufacturing process for the vaccine, but instead of formulating, filtering, and filling a vaccine, we will formulate, filter, and fill a microbiological growth media. This is in order to validate the process microbiologically and to make sure that all the components of our vaccine manufacturing facility are rendered sterile and rendered ready for the vaccine manufacturing. So the aseptic process simulation here aims to evaluate the equipment, the cleaning process, the personnel with their gowning and trainings, the materials, the sterilization process, either it's the autoclave or the tunnel or whatsoever, the sterilization equipment, also the utilities. We are going to assess all those individual components by performing and producing and executing an exact formulation, filtration, and filling, but not for the vaccine product itself. We will substitute it by a microbiological gross media. Why we will do so? Just in order for us to capture and detect any microorganism that could be present on any of the individual components of the system. And by doing this, we will able to evaluate the aseptic processing technique that we've mentioned earlier and the aseptic processing approach. We are going to evaluate it for competency for producing an injectable sterile vaccine product. Okay? Here, this is a very general process flow chart for how the aseptic process simulation takes place. It's usually that we have a powder biological gross media like the TSB or FTM, and we are going to prepare it and to formulate it in the normal preparation and formulation tanks that are to be used in the vaccine product formulation and preparation. And we are going to filter it on the same filters that are aimed to filter and to be used for filtering the vaccines. This is in order to simulate the exact process for producing and manufacturing the vaccine. And then we are going to fill, but here we are going to fill not the vaccine product. We are going to fill the media, the CSB or FTM. And after filling, we are going to send those filled units for incubation at the QC lab incubators. The incubation normally takes place at 7 days at a temperature from 20 to 25. This is to enhance the microbial growth, and then should be incubated for another 7 days at 30 to 35 degrees. This is in order to enhance the fungal growth. So by this, we are rendering the environment rich for enhancing and detecting if any microbial or fungal organisms are present on our lines. And this is to evaluate the components of the vaccine manufacturing facility microbiologically and to assess its competency for routine operation for the production afterwards of the vaccine products that should be sterile. After this, 100% inspection of all the field units after the 14 days incubation shall take place in the QC lab, and be sure that no one unit is turbid, because the turbidity for any single unit will mean that a microbial growth or a fungal growth took place. And if any turbidity or microbial or fungal growth took place, this means a failure. And in case of failure, this will mean that the process is not valid for use for the routine production of vaccines. Also, a very important point of the aseptic process simulation that we are talking about is not only to formulate, filter, and fill the media that we are talking about, but also an important and integral part is for the operators to perform the routine interventions inside the filling machine that they are assumed to perform in the routine production of vaccines. This is to assess the impact of those interventions on the aseptic and asepsis behavior of the environment, and to see if those interventions could be performed normally, or they could not be performed because the intervention itself could cause a sort of failure. Okay. And here we are reaching the last part of the roadmap towards the qualified and operational vaccine manufacturing facility. Here I'm going just to put all the individual components all together, just in a very general flowchart for you to figure out how the qualification phases are brought together in order for finally having a qualified vaccine manufacturing facility. So here in this flowchart, this is considered as phase 1. And in phase 1, our aim is to have qualified QC labs. And by having qualified QC labs, those QC labs will be able to perform the water system qualification. I don't want to deep dive into this, but finally the outcome of phase 1 will be the yellow highlighted here part, which is the water station PQ, or the water station qualification. We cannot qualify the water station using an unqualified QC lab. So phase 1 is about qualifying your QC labs in order to use them for the water system and water station qualification. And then going to phase 2, which is about qualifying your production areas. And the outcome of qualifying your production areas will be having qualified HVAC system in your vaccine production area, having qualified equipment for vaccine manufacturing, having qualified personnel for the gowning, and having qualified critical utilities like the process gases. So here, putting the output of phase 1 and output of phase 2, we will be able to proceed into the aseptic process simulation that we've been talking about, and that we've mentioned that it's normally the end stage of your facility qualification immediately before going to the routine operational phase and before releasing the manufacturing facility for operational use. So you cannot normally go for the process validation of your aseptic processes unless you've covered and you've qualified all your QC labs, production areas with all their equipment, process utilities, and personnel. So this is how we finally put all the pieces of puzzle together, starting from qualified QC lab, premises with their instruments, and qualified production areas, premises and equipment, qualified critical utilities, HVAC, water system, and process gases, either in the production areas or QC labs, and also putting the trained and qualified personnel, and finally putting the process validation either for the gowning procedure or for the decontaminating dry fog procedure or for the final end stage of having an aseptic process simulation. This is how we put all those pieces of puzzle together to finally have a qualified vaccine manufacturing facility ready for operation and use. Finally, this is a very important slide for you to know. Here we are covering the additional points to be considered while qualifying a vaccine manufacturing facility. You should be covering the cold rooms thermal mapping, the cold rooms that you will be using in storing your vaccines. Also to consider continuous and real-time monitoring for the room's temperature, which is usually between 2 to 8 degrees Celsius. And usually we put the temperature monitoring sensors in the hot and cold generated spots from the thermal mapping. Also, it's important for you to know that if you are about to use an isolator technology for filling your vaccine product, part of the isolator is to decontaminate it using a hydrogen peroxide, and an important part of qualifying the hydrogen peroxide decontamination cycle is to qualify the aeration phase by the end of the hydrogen peroxide, because any traces of residual H2O2 can cause degradation for the sensitive protein vaccines. Also, it's important for you to know that while qualifying the VHP decontamination cycle in your isolator, in your filling isolator, you shall consider to qualify it on the worst case condition, which is putting all the maximum loads that could be present inside the filling isolator, because the load here itself could act by hindering the H2O2 from reaching all the critical parts of the filling isolator, and by doing this, this can compromise the sterility and the asepsis of the environment. We can do this qualification by putting challenging biological indicators in the most occluded spaces of the load itself. Also, it's important to consider the external environment conditions and the external environment protection, because talking about vaccine manufacturing will be about talking on a hazard product. So part of your HVAC system that will be used in your vaccine manufacturing facility could be that you might have an exhaust HEPA filters on your HVAC system, and you are expected and you are assumed to perform integrity testing for those exhaust HEPA filters as well, not only for the supply HEPA filters. Also, it's important for you to test the interlocking systems that could be present on the different production room doors, because making sure that the interlocking system is functioning will make you confident of your contamination control and cross-contamination control strategy within your vaccine manufacturing facility. Here we've reached the end of our session.
Yes, John, thanks for such a wonderful presentation on facility qualification. We are actually out of time, but we might be able to take a few questions quickly.
Yes, sure.
Let me go to the Q&A section. Paul Nalokot asks that can start from Sianda. How can we have access to this guide? So we'll actually, we have shared on the chat the link to our YouTube channel, which has all the webinars that we have had previously from 2023 to now. So again, you can use that link to access this webinar. Once it's over, we'll have the link, we'll have it posted on the YouTube channel. Paul Narokot asks for clean room requalification, under which conditions at rest or in operation should grades A, B, and C be performed?
Sorry, I cannot see the question here.
The Q&A tab.
Okay.
It's down.
It's down. Okay. In the chat area, or where
The Q&A
exactly?
section. Q&A section.
Okay. Sorry, I'm trying to reach it. Okay.
Is asking if for clean room requalification, under which conditions at rest or in operation should grades A, B, and C
Yes.
performed? Yes.
Yes. Actually, we are having some tests in the clean room qualification testing that should be performed in both conditions, in the at rest and in the operational or the dynamic state. Like, for example, the viable and non-viable particle counting. Those counting for the particles, either viable or non-viable, should be performed in both conditions, in the at rest and the operational conditions for all the cleanliness grades, not only for grade A and B, which are the most critical, but only for grade C and D. So this is, for example, also for the smoke test and the airflow visualization, which is about injecting a smoke in order for you to visualize how the smoke is flowing and how it's performing inside the room, and for you to spot any dead zones within the room. Those testings should be also performed in both the at rest and the dynamic states.
Good. Now, there's also another question from John Ategeka. Thank you, John, for the excellent webinar. For HVAC qualification, what informs this qualification frequency of 6, 12, or 4 years? What major events can necessitate unplanned HVAC qualification? Thank you.
Okay, for this frequency testing, it's well mentioned in the ISO guideline 14644 part 2, and based on the cleanliness grades that we are about to qualify, we will be having the exact testing or qualification frequency. But most importantly is to figure out that the grade A and B are having the most frequent testing, for example, each 6 months, semi-annual. And for the rest, which is the less cleanliness grades, could be performed every 12 months or on an annual basis. For some testings, as mentioned, for the containment leakage and for the airflow visualization, they could be performed every 4 years or with whatever will be identified the frequency in-house. It's finally based on your in-house procedure, but the guideline has stated some strict frequencies that we are assumed to follow. That's it.
Yes. Winfred Ondieki asks if you have mentioned that the media used during APS is filtered as a form of sterilization. However, what if the media is sterilized first, then introduced to the vessels?
Sorry, I cannot see the question. Can you repeat it again? Sorry.
You have mentioned that the media used during aseptic APS is filtered as form sterilization. However, what if the media is sterilized first, then introduced to the vessels?
Okay, finally, we are aiming to imitate and to simulate exactly the same vaccine manufacturing process. So if we are going to formulate the vaccine by preparing it in formulation tanks and filtering it, so we are going to bring the TSB powder and to formulate it with WFI inside the formulation tanks and filter it on sterilizing rate filters. This is in order to imitate and simulate exactly what we are going to encounter during the routine vaccine manufacturing.
Great. So Winfred asks, you also mentioned that during the dry fog process, biological indicators should be placed in specific locations within the rooms. I could like a clarification of which form of biological indicators is used here. Is it an ampule packed or just membrane pouched spores? And if they are open in the room, what of the risk of contamination?
Okay. Usually and normally those biological indicators are strip forms, Tyvek biological indicators, in order for them to allow for the vaporized hydrogen peroxide and peracetic acid to penetrate the barrier itself of the biological indicator and to reach the spore organisms. And they are mainly located inside the most challenging and worst case locations within the room. For example, being put in the areas far away from the tank itself, or in the dead zones within the room, or the hidden areas within the room. And for the risk of microbiological contamination, finally, they are well contained in the strip carrier. So there is no risk from the microbiological contamination on this matter. So no problem in this.
Okay,
And
great.
finally, they are collected. They are collected by the end of the dry fog process and sent for the QC lab for testing and to see if the microorganisms and spores have been killed or not.
Just there are no further questions in the Q&A section. Just to ask, at what point, because the EU GMP Annex 1 recommends that we do also have a contamination control strategy. At what point should this be developed? At the initial stages or once we have qualified the facility and now it's working, then we have the dynamic contamination control strategy?
Yes, actually the EU Annex 1 has, mentioned it clearly that we are assumed to have a contamination control strategy, and this should be addressing the very initial phases of the facility and the startup phase of the facility, and it should be periodically reviewed on a periodic basis, mainly on an annual basis, for example, because by the end of each year, sort of the periodic review is to assess all the changes that have happened, the deviations, the trends, and so on. But if any changes have happened during the year itself, the update for this contamination control strategy could be performed on spot. We might not need to wait till the end of the year, but finally, the update should be performed at least on an annual basis, okay? And since you've mentioned, yes, it's a dynamic document, so any changes that might have occurred on your facility, on your process, on your equipment, and so on, should have been reflected on spot and instantaneously by updating the document on spot, not to wait till the end of the year.
Great. We are 10 minutes past our time. So I could like John, maybe you could make your final comments, and then we close this up.
Finally, I would like to thank you all and hope you took the most of benefit from this webinar session. I was so glad attending with you and presenting to you this session, and actually looking forward to having much more opportunities with you, and would like to thank also KEMRI-Wellcome Trust for this great opportunity, and would love to maybe to keep my contacts with Clive, and he can share them with you for you in order to ask me on any occasion, in any time, about anything. I will be always here for you at your disposal.
Yes, kindly you can use the chat ask any questions and we'll respond even after the closing of this webinar session. Thank you very much to everyone in this webinar. We could like to hear about your experiences and what else you could like us to discuss in upcoming forums. Before we close, kindly requesting that you take a few minutes to respond to the post-webinar survey posted in the chat. So there is a link on the chat tab for you to give us feedback. Have a nice day and looking forward to more engagements with you. Thank you.
Thank you. Thank you so much. Thank you all. Have a good day. Goodbye.
Thank you. Bye.
Goodbye.